Good afternoon, everyone. Welcome to the presentation of our EU project Valuable, which is within the context of a circular bioeconomy. And I'm welcoming our guests here in presence. Our friends from Innsbruck, from ADSI, our project partners, Evonik, KAIMA, GST, and of course, Mr. Weyer from the Bavarian representation of Bavaria. Welcome. And our project coordinators, Marion and Michael. And it's my pleasure today to present the outcome of our valuable project, which has taken now 3 years of commencement to bring a circular bioeconomy forward with multiple project outlets and real products that end up on the market. So what is valuable? Valuable is the valorization of fungal biomass using novel enzyme technologies. And today I would like to start with our motivation. Why do we do this project and how does it fit into the context of a circular bioeconomy in terms of the political outcome that we want to achieve with the technology that we are preparing? So I'll first talk about UN sustainability goals and how we can achieve them, then the outcome of Valuable, and finally our summary. And of course, what you see here is the technologies that we deployed to make a concept into reality. So basically, we have a still growing world population, and it's expected that we reach 10 billion people on this planet by 2050. An increase of about 50% over today. And the greatest growth will be in urban areas, particularly in Africa and Asia, putting immense pressure on our infrastructure, food systems, and healthcare. And with that, we also have linked issues to feed this population and to enable sustainable growth. One of those will be water shortages. So we anticipate that global food production will increase by 70%, and already 2 billion people experience moderate to severe food insecurities, particularly in arid and urban areas of this planet. 40% of the global population is already affected by water scarcity, and the number is still rising. We've seen that recently with increasing temperatures, that we have failure in crop growth that affects food security as well as the supply of potable water to our population. And one of those effects will be drought and hunger. Climate change is accelerating the frequency of droughts, leading to crop failure and food crisis. And already the IPCC warns that up to 180 million additional people will face hunger each year, with temperature rising beyond 2 degrees globally. And with rising temperatures, there's also an influx of diseases that are common to our areas of the planet, particularly here in Europe, where we used to have moderate temperatures. We now see the influx of tropical diseases such as malaria and dengue, not only through increased travel, but also through increases in temperatures, and particularly things like the tiger mosquito are taking hold in our ecosystem, which endangers really our population, but also the biodiversity in Europe. So these are points to address in the future, and really where we want to go is into a more sustainable growth, even under extreme conditions, meeting these 17 UN sustainability goals. Zero hunger, good health, quality education, these are things that we can address with Valuable. While we work also in academic settings, gender equality is something that we are addressing constantly, as well as climate actions, life underwater and on land, as well as sustainable cities and industry innovation. All of these sustainability goals can be achieved by using biotechnology as a key technology to achieve a circular bioeconomy. So how do we actually deal with the challenges that we have to date? So one of the biggest challenges that we have is plant oils that are something called a first generation feedstock for a sustainable economy, but they're associated with land use change, monocrop agriculture, and deforestation, having significantly negative effects on biodiversity, with the orangutan being one of the iconic species in tropical regions being affected by these monocultures. On top of this, of course, here in Europe, we have rapeseed cultures, also monoculture, heavily impacted by pesticide use, which negatively impacts particularly insect biodiversity, negatively affecting pollination and crop yields. And we try to address these issues that accumulate into food insecurity by using a waste stream of the food and biochemical sector to achieve the production of oils using a new biotechnological oil production combining enzymes that and other biocatalysts that have low environmental impact, that are fungal derived and show low allergenic potential, all to produce application derived intermediates that impact food sectors, the biofuel sector, oleochemicals, and the polymer sector alike. Moreover, we also have a side stream that is chitin, and commonly chitin is produced from crustacean shell waste, and to achieve the release of chitin and chitosan as the active ingredients, you use toxic mineral acids that lead to environmental pollution, and you have a product that is animal derived. So chitosan that we buy today and that is used in pharmaceutical applications, waste treatment in cosmetics and personal care is something that does not fit with the clean and eco labels that are particularly manifested in the cosmetics and food industry to date. So what we addressed is the production of chitin and chitosan using biotechnological means, where we did waste recycling using fungal biomass and combined that with oil producers and lipid producers to produce chitosan and lipids for particularly the cosmetics and personal care sector, together with adhesives and material sectors. So a little bit on the Valuable project itself. This project had a runtime of 3 years, and we've been operating since 2023. And this project comes to a close at the end of this year, and it was funded with 6 million euros by the Horizon Europe call. And we had multiple partners from several countries, SINTEF in Norway, NMBU, also a Norwegian university, Evonik, one of the big industry partners, also a leader in biotechnology. We had small innovative companies, Global Sustainable Transformation, focusing on sustainable lipids, Biolog Heppe, that is focusing on chitosan production and is in the market currently with crab shell waste-based chitosan, and is very interested to make this transition to more sustainable non-animal products. And of course, Kaima from Thessaloniki, Eleftheria being here as the representative, focusing on wood board manufacture and sustainable adhesives. And alike, we also have Megara resins. I think Yannis is online to give a presentation on there. Also on adhesives and bonding materials, and of course, our partner from Spain, Idenir, who's concerned with the dissemination of the project content. So what did we do technologically in this project? We converted a waste stream from citric acid production into value-added products using a holistic, waste-free biotechnology approach. And why did we choose residues from citric acid production? It's one of the biggest biotech products that is produced globally with 2.5 million tons yearly. And citric acid we find everywhere in cosmetics, food, pharmaceuticals, and also in the material use. So there is a broad usage of this, but there is a production that is based on this organism, Aspergillus niger, a filamentous fungus. And this fermentation converts sugar into biomass and citric acid. So while citric acid is the product that is being sold, the leftover biomass of fungal mycelia, as we call it in the technical terms, is a waste of this rather large industry. And it amounts to about 600,000 tons a year. And currently, it is sold into the feed market as an additive, particularly in piggery feed. One of the problems with this is that this biomass contains a high content of nitrogen compounds derived from DNA, and that leads to problems with digestion in pigs and potentially kidney damage if you use too much of this in the feed product. And since the meat usage and pig farming is actually declining in Europe, we are left with this waste with no value to the industry. And there are two big citric acid producers, one being in Austria and one actually here in Belgium. Yeah, that's the two big ones. And they're producing together over 1 million tons of this waste a year. So we thought about using this waste and turning it into useful products for various sectors. So starting, we had this residual fungal biomass, and we developed an enzyme system that solubilizes this solid waste into a fraction that's rich in chitin and rich in sugars. The chitin-rich fraction was then converted into chitosan, which is the high-value outlet of chitin with applications in adhesives used by Kyma and cosmetics. Yeah, this is the Evonik outlet. We then took the sugar-rich fraction and did an advanced precision fermentation to convert the sugar into yeast oil. And the yeast oil we then tested in cosmetics as well as in resin applications. And the composition of this yeast oil is similar to palm oil. So it actually is a one-to-one replacement of plant-based oils. Yeah. And one of the hallmarks of this process is that we have total nutrient recycling into the fermentation. So there is no waste streams. It is a completely waste-free process. So having these products at hand, we then did first demonstration activities that are close to the products. We used the chitin to form adhesives that then were tested in wood panel production. And actually, I think Eleftheria will tell us a little bit more on this. The wood panels produced by these new adhesives were not only completely bio-based, they also had better performance to the standard resins that are normally used in production. Plus, they had one other benefit. You had less organic outgassing. This is a side stream, basically, of wood panels. So it's also more environmentally friendly. So on the other hand, we use the chitosan for devising new cosmetic applications that are anti-allergenic and comply with green and clean labels. And on the side of the yeast oil, these came together to basically substitute plant oils with our yeast oil applications. And these together were synergistic to form new applications in the cosmetic market. The yeast oil was also tested by Megara in resin formulation to produce sustainable resins. And as you can see here, these resins are actually also performing equivalently to the plant oil-based resins that are already on the market, and we can replace basically the plant oils in these formulations with our yeast oil. So there is a direct product outlet, and these products are now ready to be released to the market. Okay, now a little bit on the partners. So who was doing what? So we had NMBU, the Norwegian University of Life Sciences, with a specialty on enzymatic hydrolysis of Aspergillus niger biomass and the modification of chitin. Biolog Heppe in Germany, who does chemical modification of fungal chitin to generate chitosan. Megara Resin, the production of sustainable resins from yeast oil, and Kyma, that produce adhesive formulations integrating chitin and chitosan derivatives. Ourselves, as the coordinator, we were also focused on oleogenous yeast fermentation based on Aspergillus niger hydrolysate, and we achieved that not only at lab scale, but also technical scale up to 1,000 liters, while startup Global Sustainable Transformation dealt with the yeast oil separation and purification and application development. Evonik took on the role of testing these new ingredients in cosmetic formulations, while Idonea, our Spanish partner, dealt with communication, social media representation, but also was focusing on process modeling, process assessment, and exploitation. SINTEF, as the overarching partner, was doing process modeling, techno-economic validation, and assessment of process viability within economic models. So what are the stakeholder benefits? There are both benefits for the green and yeast oil in the cosmetic and resins industry. They are green feedstocks, they reduce the environmental impact, improve public perception, and open new market opportunities, particularly as they are vegan products. So in cosmetics, that is important to address a new labeling system as well as the consumer needs. On the other hand, we are dealing with releasing citric acid producing companies from their waste. Yeah, so we are increasing the value of this waste. Normally in pig feed, about 300 euros per ton. This waste is generating value. If you now sell yeast oil, it's 1,000 euros a ton, and chitosan sells over 20,000 euros a ton. So there's significant value increase compared to the classical outlet of this waste material. So, and then we are already at the summary. The biotechnological production routes meet UN's sustainability goals and climate action, create value-adding sustainable products with better performance from waste streams. In terms of the oleogenous yeast, we have a waste-free transformation of biomass residues into food and cosmetic products. The yeast oil is a better sustainable route for food and cosmetic performance additives. And we have environmentally friendly and economically effective production routes with products entering the market after this project is finished. Yeast oil in particular can replace both cocoa butter and almond oil in food and cosmetic applications. Chitosan can supply the market with anti-allergenic and vegan alternatives, and they increase the supply chain resilience and raw material independence of the associated producers, which is very important to date as we are entering a new era of global competition. And with that, we look back, achieving actually quite a number of UN sustainability goals. We have global health, learning, gender equality, supply chain resilience, industry and innovation, support, responsible consumption, climate action, life above and below water. So quite a number of the UN sustainability goals could be reached in this project. And if we look back at all of this, we should also think about how do we translate these innovations that came out of this project into something that is adopted by the market and is used by consumers in the European Union. Yeah, basically, our concern in the next step is how to convert the money that the EU spent on this technology development into job creation, the securing our competitiveness as the European Union, so not only Germany or Norway. It's really about cooperation and how we bring this technology then from Europe to other locations globally. Yeah, so really this translation industry is important because larger companies like Evonik not only operate in Germany or Europe, they have hubs all over the globe. So if they adopt the technology, then that means there's also access to other consumers, which will increase the chances of having a circular bioeconomy on a global scale. So really this translative aspect is very important to us. And I would like to open the discussion later on how we act on policies to bring this technology into the market and beyond. Thank you. And now we are leading over to our partners. And I think first up is Eleftheria is an R&D support manager and IP protection manager at Kaima, chemical engineer with an MBA in business administration. And since 1996, she's part of Kaima as a company, making her an expert in the field of chemical engineering in the adhesives and wood board manufacturing. Kaima itself is a pioneering Greek company specializing in the development and licensing of innovative chemical technologies, primarily in the field of wood adhesive and resins. The company has established itself as a global leader in sustainable and eco-efficient solutions for the wood-based panel industry, and Kaima technologies contribute to reducing formaldehyde emissions, very importantly, and improving the environmental footprint of industrial production, aligning with EU green policies and global sustainability. Looking forward to your contribution.
Thank you. Thank you for the introduction. Good afternoon, ladies and gentlemen. Already the introduction was very well given, so I will. Okay.
But next to the table, then people can hear and see you.
Okay. Thank you. Himar is a chemical company. We are developing and supplying technology for adhesives and binders and the chemical additives, which are applied in the manufacture of wood-based products. We also offer technical support and training services to the adhesive and panel manufacturers and R&D services for third parties, and have a strong participation in research projects. We have also some specialty services like provision of chemicals upon request, equipment representation. Our lab is accredited to provide certificates for formaldehyde emission from wood products. And also we perform resin additives and board testing and evaluation for our customers. Our technology is actually based on polymers and resins based on formaldehyde, urea, melamine, phenol, and their combinations. But we have a strong portfolio of technologies for bio-based adhesives and no added formaldehyde binders. All these products are used to manufacture fiber boards, particle boards, plywood, laminates, and engineered wood products, as well as insulation materials. And we have a focus on safe and environmentally friendly products and technologies. Himar has a market share of 10% of the global particle board and fiber board production. These products are produced annually worldwide with our technology. We have, as already said, we have a focus on green technologies and products, and we are in the business since 1977. And some words about the wood adhesives. Wood adhesives are actually polymers, polymer resins that are combined with chemical additives and are normally derived from fossil resources. Chemicals like formaldehyde, urea, melamine, phenol, and resorcinol. These polymers, when synthesized, they are blended or coated or applied on fibers, on wood fibers, wood particles, wood strands, veneers, or even lumber. And they are then formed into final products, the wood panels, which are used to make furniture and also to make construction parts like roofs, decks, floors, kitchen frame, kitchen tops and frames, and things like, and also furniture in the house. The bio-based adhesives actually are derived from materials and chemicals that are coming from renewable resources, from natural products or other bioprocesses, and also they may are derived from cellulosic waste. They are trying to reduce the environmental footprint of this part of the industry, which is apart from the chemicals, they are using wood as a main substrate. So if we replace the resin, the polymer, with a bio-based adhesive, then the total product will be more environmentally friendly. The estimated market growth rate for bioadhesives is for the period 2025 to 2033 is 11.7%. So you can see that there is a great potential in these products. Invaluable, Himar has received several samples from chitosan, chitin, and yeast oil, and from the partners, Biolog, NMBU, and TUM, and has used all these samples as reinforcement agents in phenolic and urea formaldehyde adhesives for the production of plywood and particle board type panels. The panel properties were evaluated and compared with the reference, which were actually fossil-derived adhesives. Also for comparison, we have used chitosan samples with crustaceans that were provided by partner Biolog. The positive effect of the use of chitosan, chitin, and yeast oil from viable project was actually proven in the performance of these adhesives when producing these board samples. Moreover, we have developed chitosan-based bio-based adhesives, totally using chitosan from a valuable project and other bio-based feedstocks like monomers, crosslinkers, and additives that were not coming from fossil resources. The panels that we produced, upon evaluation, they showed that they had properties that were equal with the conventional type adhesives, thus proving the path to the market adoption of these products. Some pictures from our process when we produced plywood samples, where we apply the glue mix on the wood veneers and then we press them so that the resin is solidified and we have the final product. And also we produced some samples from the project. We prepared actually stands for your mobile phones. And that's all from me. And thank you. Thank you for the collaboration in the project and also for your attention.
Eleftheria, thank you very much. I think we can actually talk about the market opportunities of the products later on in the discussion. Also, since we actually have some representative of the EU here, deregulation and what you think needs to be done to bring these innovations to market. Yeah. Also from a sort of medium-sized business point of view, that would be very interesting to hear. Okay. Thank you very much. And now we have Mahmoud Masri as the next speaker. So about Mahmoud, he's the CEO and co-founder of GST. For his work on cultivation of oleaginous yeast, he earned his PhD with summa cum laude at TUM 2019. And GST is a spin-out of TUM, and Global Sustainable Transformation sees itself as a technology provider and developer of clean mission to turn waste into value, particularly through the proprietary FHCR platform, which transformed biogenic industrial waste streams into high-value oils, bioactive compounds, and bio-based materials. And Mahmoud's leadership enabled GST to grow from a university initiative to a startup that bridges scientific excellence with real-world applications, which actually happened in this project. Mahmoud, the floor is yours.
Thanks. Good. Thank you for having me today, and thank you for attending. I will use this 5-minute chance to introduce GST and what we did also favor the valuable. Yeah, as Thomas mentioned, GST is very small company, young company, but with big vision. And hopefully we have high impact on the sustainability transformation. How we do this, why we have this, impact, actually that's coming from our product and how we produce product. So easily we have palm oil equivalent alternative, cacao butter equivalent, and sunflower oil replacement. These 3 products has not been produced from any agricultural land, from any plant. Actually, it's produced from one fermenter in the lab where we are using no agricultural land. Why we need this product and what the motivation behind this product? Actually, we are trying to create a new platform or new generation of material that can replace plant oil without depending on land use change or impacting the environment or deforestation. Actually, we were going to start from our side stream, locally available side stream, and with negative or low carbon footprint, and then we can have a market accessible product. So, as mentioned, Thomas mentioned, how we do this. We have over the last 12 years together with Thomas and with over in TUM, we have created what we call it FHCR platform, which is starting from any side stream and start for fermentation using a unique fermentation system. And that's allow us to have high productivity. Then instead of to separate the oil and extract the oil, instead of using high pressure homogenization or high pressure mechanical stress, followed with toxic chemical extraction, we using purely enzymes which are produced by ourselves. And this enzyme allows to hydrolyze the whole cell wall. And then by centrifugation, we can separate directly pure oil and recycle all what is not product to the next fermentation cycle. That platform was applied partially in valuable, but the role of GST was focusing on the enzymatic hydrolysis and in the centrifugation. So we get the culture from the TUM, thankful, and then we give them again the water phase to check the recycling of that product. The main task for us was invaluable, how to scale up the lipid recovery, that means optimization, the centrifugation together, combination with the enzymatic hydrolysis. To do this, also, we need to produce our own enzyme, which are our proprietary enzyme, IP enzyme, which we have also upscaled this enzyme production and optimized it further. Then, after we recover the oil, we start to formulate it. Actually, the 3 products which you saw them at the beginning of the product has been partially optimized and focused in the way how to meet the end user application and the standard specification for that product. During this project, we could increase the scale of the downstream processing, which is usually always in the biotech. The downstream processing is the key to scale up any technology. So during that project, we scale up this downstream processing. We have reduced the energy consumption for the downstream processing, increase the efficiency, and also reduce the total cost of the downstream. This project allows us to move to the next stage. Now we are thinking about going how to do it in the demo scale, how to go for 100 cubic meter fermentation, or how to start production in the yeast oil, not in kilo scale, but actually in ton scale. And that's in the next growing of the GST. We're always looking for new partners to have all this way back to scale up and to access to market. And we always offer our partners to attract new customers, to have access to good oil and fat, and then to have achieved supply food chain or supply chain, and also improve them carbon footprint. That's GST. That's our team. And thank you for listening.
Thank you, Mahmoud. And particularly looking at the products and how we look at placing them into the market to replace plant oils is something to be discussed.
Yes.
Yeah, that's one of the biggest hurdles we face. Yes. Yeah. Okay. The next one is Nadina Drukan. Yeah, she's from Evonik. And Evonik is a specialist in biotechnology, but also, of course, with an application focus on cosmetics and high-value products. So Nadina earned her PhD also at the Technical University of Munich, but is an organic chemist by training. And since 2021, she's technical manager at Evonik in Essen, and where she is a part of the applied innovation team focusing on cosmetics and particularly hair. Those yours. Thank you.
Perfect. Thank you so much for the very kind introduction and a warm welcome also from my side. I'm very happy to be able to be here today. And yeah, so in the next, 5 to 10-ish minutes, let's see how much time it will take. I would like to briefly introduce Evonik to you, as it might not say something that everyone is already familiar with. And then I would like to highlight the tasks, but also the achievements that we have done within the EU Valuable Project, and then introduce the capacities, but also the methods that we have used to evaluate different samples or materials received within this project. So the history of Evonik, I think the name Evonik is something that maybe rings a bell to some of you. I think usually when you think of soccer, we were sponsoring Dortmund for many years. I think this is something that most of people know Evonik from, but actually the knowledge of Evonik roots back almost 170 years. And the branding or the name Evonik used in 2007, but it unites different areas of expertise, which you can see on the bottom left. So for example, Goldschmidt, but also Hüls, and also Ruhr. So there are many companies that merged into Evonik. So it's based on very different areas on expertise. And just so we know, or just to give you an example on how we looked back then, this is the Chemische Fabrik Theodor Goldschmidt in Essen. It is still where we are today. So this is the headquarters in Essen still. And this is actually also where I am currently located. Yes, so the organization after all is has different divisions, but also business lines. So at the moment we have 5 different divisions. You can see that nutrition care is highlighted. This is because this is the business line I am working for, more specifically the Care Solutions. And this is also the part that was contributing to the EU Valuable Project. So different colleagues and also divisions have contributed into this project, R&D facilities, but also our skincare colleagues, and as Thomas introduced very kindly, also the hair care and cleansing department. what is also very nice to see, I think, is that, we act, of course, as a global company. And on this slide, you can see different production sites, but also business R&D and application technology centers. So you can see that we are based in North America, but also South America, EMEA, and also Asia. And depending on the dots, on the color on the dots, it is either a production site or application technology centers. So you can see that we have different, global locations, which is very nice, because as you remember, maybe Thomas in the beginning said, if we introduce a raw material into the market, then it's very likely that we try to introduce it globally. Yes, and now I would like to introduce what we have done for the EU Valuable project. And in particular, we have looked into the yeast oil samples, but also chitosan samples. And maybe to give you a little bit of background why we are interested in these kind of raw materials. So as you know, there are certain trends in the market driven, for example, by sustainability. So we're always trying to replace the raw materials that we are currently using with more sustainable raw materials. For example, if there for example, for the yeast oil, if you think of palm oil or palm kernel oil, this is something that many companies are trying to replace. So this is something that we, of course, and also other companies are looking into. And for the chitosan, I think this was also introduced very nicely. If you think of any vegan claims, so the chitosan currently introduced in the market is not vegan compliant. And this is also a very big trend in the cosmetics market that many customers are looking into. And I think the cosmetics market is very emotionally driven. So the end consumers kind of give us always the direction that we have to head into. And we are very happy that we were able to be part of this EU project, because it gave us a lot of insight into alternative raw material. And also we could learn a lot how we have to adjust the samples or the properties of the samples for the properties that we need for certain applications. And the work packages that we were focusing on were to, let's see on the left, the yeast oil. And this is what we have done within the work packages. So we have evaluated the physical chemical properties of the yeast oil samples. We have introduced this into the emollient chart. This is something which I will explain on the next slide. We have also prepared different emulsion-based formulation. We have checked the stability of these formulations. We have compared the sensory properties. So usually the consumer very likely will see if you change a formulation. For example, if you use a body lotion for many, many years, and then when one raw material is being exchanged, you will feel that, for example, it's more sticky, it's more whitening. And this is something that we try to target from the beginning to see the impact of certain properties of raw materials in the overall formulation. And so this is something which I also explain on one of the next slide. And for the sensory properties, or in this expert panel, we also focus on appearance, olfactory properties, for example. And what we have also done is, and this is how the yeast oil can be seen. So either you can use the yeast oil as is, exchanging it with raw materials that are currently used in the market, naming, for example, olive oil as an example, or you can split the yeast oil into the single fatty acids and then convert it to derivatives. And this is also one source of fatty acids that could be a potential outlet in this project. And this is also something that we have, I would say, successfully done. And for the chitosan and its derivatives, we have evaluated the solubility of these raw materials, also with support of the partners in this project. We have evaluated the conditioning properties of single raw materials. So usually, if you think of a conditioner, after using it, your hair is very soft. And this is because most of the conditioning agents interact with the damaged hair and give you a very soft feeling. And for the chitosan, we expected this property because it's pH dependent, and at certain pH values, you have a cationic raw material. And this is why we expected it to be or to work as a conditioning agent. We have also evaluated the styling properties We have focused on film forming, curl retention, water uptake, and 3-point bending measurements. I will go into detail on these methods on the next slides. The results that we have received here were also very interesting to see. And we've also checked the formulation compatibility. So this is always the next step. So first we check the properties of every raw material, and afterwards we see if we have stable formulations depending on the formulation that we want to achieve. Either it's a shampoo formulation or a body wash or a cleansing gel. And what we always do is we compare the samples to suitable benchmarks. This might be either raw materials, but also final formulation. So it depends if this is a customer project, for example, or if this is really focusing on single raw materials. And then something which is also very important for us is really the biodegradability of these raw materials according to the OECD guidelines. And this is something that we have also done the testing externally for the samples that we have received, especially for the chitosans and the derivatives of the chitosan that we have received within this project. Yes, for the yeast oil, I would like to just give you an idea of what we are doing at Application Technology. So it's very, very close to what end consumers will also feel. And we try to evaluate or develop methods that focus on certain properties. And just as I said in the beginning, if you use a body lotion and you feel it's sticky, you won't buy it a second time, I would say. And this is something that we try to find out in the beginning of the product development, either for the single raw materials, but also for the final formulation. And for that, the skincare colleagues, so this is the contribution by the colleagues of our skincare department, have developed the Evonik emollient chart. And what is depicted here is really the surface tension and the viscosity. And depending on those two parameters, you can classify different emollients. And this is always very helpful to us, but also to our customers, because by giving them or sharing the emollient chart with them, they know what to expect. So usually they know 1 or 2 emollients already. And then after clustering it, they can see, okay, what can I expect from this kind of raw material? It might be if they look into the spreadability. So the bigger the circle, the higher the spreadability of this material. Then if you look at the color of the circles, it gives you an idea on the polarity of the raw materials and the outlines. In this case, for the yeast oil, for example, we have a fully vegetable-based one. I think this was one of the first sample sets why we have classified it as fully vegetable-based. And then if you compare it to olive oil, for example, you see that in this case, we would expect very similar properties and maybe also similar to number 18, 20, 21, and 22. And then, as a customer, you can check, the emollient, which the properties are very similar to. For the sensory expert panel, which we have also done for the EU project, this is like a rheological and sensorial behavior. We have an expert in-house panel, so really trained people that evaluates the formulations that we are developing, sometimes also only single raw materials. But I think as every raw material gives a certain impact on the overall formulation, usually we use really final formulations. And here you can see the properties that we are looking into. So really spreadability, whitening, oiliness, waxiness, absorption, and also certain properties after 5 minutes. And here you can very nicely see. So these are just dummy formulations. But if you, for example, take a formulation without a certain raw material, then you can really see the differences. And sometimes it's really surprising how big the impact of a single raw material can be. So this is something that we have done for the yeast oil, comparing it to raw materials that are usually incorporated into water and oil, but also oil and water formulations. And last but not least, I mentioned it very briefly, the further conversion of the single fatty acids. So we have split the yeast oil externally into its single fatty acids and converted it to two derivatives. So one is the partial esters, which can be used also as emollients. And here you can see just exemplary. And the other one is the conversion with alkanol amines to emates. And these are very often used, for example, as foam boosters or rheological additives. So this might also be a potential direction of these single fatty acids and further derivatization. And both of them, I have to say, were also successful. So it was very nice results. And supported, I have to say, by our R&D colleagues, of course. And for the chitosan, I wanted to start with a market example because I think it shows very nicely that this is a topic which is still very prominent in the cosmetic market. You can see that these are the two. Yeah, so Dyson was not prominent in the hair care market until those two products launched last year. So you can, or I hope you can see that they really use chitosan as a key raw material in these formulations. Even though if you look at the Inky, it's a bit longer, but still I think it's very interesting because very rarely, if you look at shampoo packages or styling products, you never see a key technology on the product and packaging. So usually you see hydrating, moisturizing, glowing, whatever, but you never see really the technology on the packaging. So I think it's rather unique. It just shows that this is a technology which is very interesting to the cosmetic market. And I think, and I also want to highlight maybe that the chitosan, which is used for Dyson, is derived from mushrooms. So a little bit of another source comparing to what we have used in the EU project here. And yeah, on the next two slides, I would like to talk very briefly about the methods that we're using for the evaluation of styling properties. So to the left, you can see the curl retention test. It's a very easy test, I would say. I think everyone exactly knows what is happening here. So you look at the curl length at a certain time, and after 2 hours at a very high humidity, you look, you measure kind of the droop of the curls after this time and determine the hydrophobicity of the hair. So the more, the less the curl length is after 2 hours, the better the hydrophobicity given to the hair by the styling raw material. So what you want to achieve is by using a styling product is to increase the hydrophobicity of your hair. So this, whatever your hairstyle is for today, it should last as long as it can. And this is given by the hydrophobicity of the single raw materials. And another method that we are using is the texture analyzer, or more specifically the 3-point bending. And I hope you can see it, but here you can see a hairdress, hopefully, maybe even better in that picture. But usually we take a hairdress, we dip it into a solution, which is, for example, 1% raw material in water. And then ideally, and this is only possible if a film formation occurs, we measure the force that is needed to break the film on hairdresses. So the hair is really hard and crispy when the film formation is very occurs. So this is a very nice method to see how strong the film formation is. And this is can be compared to different raw materials, benchmarks, and also different samples within this project to see what impact different properties of the single samples have on, for example, the 3-point bending. Yeah, and last but not least, we have also evaluated the conditioning performance. Usually we do that by the sensory hair swatch test, which you can see in the middle. So this is kind of a professional combing of hairs. And we also have an expert panel for that in-house. So sometimes it's very hard to really distinguish between two samples. So you have to really train that. It's not as easy as you think. I think what you can always do is, yes, conditioning, no. But if you really have to evaluate or see small differences, it can be very tricky sometimes. And we evaluate these properties on wet hair, but also on dry hair. So we comb through them, we see how easily it detangles, how easily the combing goes, and how it feels. So does it feel soft or does it feel greasy? Do you hear something on your hair? And the same happens for dry hair. Though I have to say that the wet properties are the more important ones, because here you can really distinguish between different properties more easily. And I think it's also the more important one, because once you're under the shower and you use, for example, a conditioner, you will immediately see if it's not going to work. On dry hair, it's easier, because on dry hair, you just comb through it and then you forget about it. But under the shower, I think it's always something that you will remember. And there are other methods as well to see. So to the left, the combing force. it's a determination of forces needed to comb the hair. So it's very similar to the sensory hair swatch test, but I would say maybe more analytic. But usually we only use the combing force if we already know that the raw material is kind of conditioning, because otherwise it can be very tricky. You cannot comb through the hair, it stops all the time. A lot of frustration shared by our colleagues already. So this is only something that we do complementary if we know that the conditioning and properties are given. And then repeated combing. This is something that we have not done for the project, as this device is only available in Brazil. But you can do really repeated combing, and that means really 1,000, 2,000 times. And then you can count how many hairs broke through this combing process. So you just count the hair that is lying on the bottom of this device, and it gives you an idea how well your product protects your hair against hair breakage. And we have also other methods available, which I'm not mentioning, and we also have not covered this in this project, but for example, heat protection, hair strengthening, wash fastness, anti-static loss, volume, or the hydrophobicity, where we are using also a tensiometer, which is also a bit tricky. Yeah, and with that, I hope I could introduce a little bit what Evonik has done without going into too much detail on the results. If you have any questions, of course, feel free to ask them in the Q&A. But yeah, other than that, I want to say thank you for your attention.
Yeah. Nadina, thank you very much. Actually demonstrates that chemical equivalence of the main component doesn't mean equal performance. That the side products at times are important to meet the technical challenges of performance in a product.
Yeah.
So maybe we can make this. So as our last speaker, we have an online contribution from Megara Resin. So Yannis will talk to us about their contributions. Yannis received his BSc in chemistry from the University of Athens. He completed the PhD at the same university and followed as a postgraduate studies in inorganic chemistry and catalysis. During his academic research, he gained experience in the synthesis and characterization of metal complexes and various ligand heterocyclic carbenes, and studied their catalytic activity in organic synthesis, including utilization of CO2 in the synthesis of organic compounds. He worked with several national and international funded research projects, and since 2020, he joined the Research and Development Department of Megara Resins. During this period, he has studied the properties of composite materials and gained experience in the synthesis of a range of polymers for coating applications and their combinations with carbon-based materials and the properties of new composite materials. So, Yannis,
thank you for the introduction, Professor. I will present you some information about Megara Resins. Megara Resins was founded in 1961 by Anastasios Fanis, and for nearly 60 years create new innovative technologies to help coating formulation meet their customers' more demanding applications. Megara Resins is a privately owned resin manufacturer and supplier of raw materials for industrial and architectural coatings, rosin-based and other synthetic resins for the paint, adhesive, paper, and construction industry. Today, Megara Resins is regarded as the most innovative Greek supplier to the coatings industry through its continued investment in research and development, technical support, and new product development. Megara Resins retains a key position in the Greek market with 3 manufacturing sites, state-of-the-art research and development center, while its business extends globally with strong presence in over 56 countries. For the year 2024 achieved sales of 98 million euros with net income of 5 million euros after taxes. The product portfolio includes powder coating resins and additives, rosin derivatives, liquid resins for coatings and inks, paper chemicals, emulsions, and UPRs, unsaturated polyester resins. A more analytic catalog we can see. We have carboxylated polyester, hydroxylated polyester, cross-linkers, rosin derivatives, rosin esters, maleic resins, rosin phenolic resins, hot metal adhesives, paper chemicals, liquid resins for coatings and inks that include alkyd resins, solvent-free alkyd resins, urethane modified alkyd resins, solvent acrylics and waterborne acrylics, emulsions, and unsaturated polyester resins, acrylic dispersions, copolymers, and homopolymers dispersions. Our role in valuable projects. We received sample of yeast oil with main target to produce the corresponding alkyd resins. So we demonstrate the successful development of alkyd resins from yeast oil and the validation of their suitability for surface coating applications. Establish potential for replacement of traditional solvent-borne alkyd resins from palm oil with waterborne alkyd resins from yeast oil. Our main achievements. We demonstrate that yeast oil can be successfully applied in the synthesis of alkyd resins. The combination of yeast oil based alkyd resins with isocyanated cross-linkers proved that provides coatings with excellent properties. Yeast oil was successfully combined with other vegetable oils for the production of alkyd resins. We developed a protocol for the production of waterborne alkyd resins using the reverse phase techniques, anionic and non-ionic surfactants, but with poor properties as coatings. A promising alternative is waterborne urethane modified alkyd resins for coating applications that provided coatings with very good properties. As future prospects, it would be nice if we can have yeast oil with higher content in unsaturated fatty acids, and for the production of alkyd resins, it's critical to have a competitive price per kilogram, and if this can be achieved. So it was a short presentation. Thank you for your attention.
Yanis, thank you very much.
Yep.
And with this presentation, we actually will finish the technical goals
And
this, and I would like to thank all the online participants for listening to the contributions. And I think what we'll do, we'll first have some coffee and then discuss the political aims in the small round here in presence, because we have a unique setup now. I think we have the representation of the CSU in Bavaria. And then we have actually a legal expert from the EU Commission that is dealing with innovation. and we can actually look at what is needed to transfer this into the market and how can we alleviate the barriers to innovation in the EU. I think that is very important. So for you, all this technical surroundings are probably not so interesting. The interesting thing is what can we do politically? And for this, framework conditions need to be discussed. Also, what is the sort of quality control that needs to be done to maintain a label in the market. For this, we have ADSE actually here that do that frequently for very large industry clients. And I think we will have a vibrant discussion after the short break. And with that, I would like to thank all the online participants, and we'll close this session. Thank you very much.